Hello everyone, this is Professor Thayer, and today we're going to talk about hidden Markov models. So let me share my screen and let's get started. So we're going to talk about a topic that is very important. It is about how we can make some probabilistic models that are going to allow us to capture patterns in a probabilistic way. Hidden Markov models are one of many machine learning tools that are possible to use, but they have some very special properties that allow them to be especially good at this task. So we'll be talking about that today. Let me just swap my settings here. Display settings, swap, and hopefully you'll be able to see my slides. There we go. All right, let's get started. So here's an overview of what we'll talk about today. We'll begin with an overview of hidden Markov models, what they are, and a little bit about how they work. Then we're going to talk about hidden Markov models and probabilities, and to this we'll have a special treat. We'll be visiting the slightly dishonest casino to understand how that works. And then the third part, we're going to be talking about a hidden Markov model primer, where we talk about three major areas. That is the architectures of hidden Markov models, how you can use them to train, and how you can use them to score. Finally, we'll take a look at some sample results from some of my previous work, just so that we can see how hidden Markov models might be applied to actually interpret data. Hopefully you find this interesting and helpful for understanding how hidden Markov models work. So, to begin, what is a hidden Markov model? It's a statistical modeling technique that uses machine learning to identify patterns which are allowed to arise with many variations on a central theme. We can use this for proteins or DNA, and it's a means of addressing a bioinformatics problem, among other things. Hidden Markov models were originally brought up through Bell Labs. They were initially used for voice recognition, and this was way back in the 1970s or so, when people were just starting to try to work out how voice recognition software ought to work. The idea behind the problem they were trying to solve was to recognize a word from the sound information that someone would get. So for example, imagine having a microphone and having each person in a classroom speak the same word into the microphone. So maybe, for example, the word milk. And so you might pass it around and you might get some variation in pronunciation because people pronounce things differently. And so the idea would be to recognize the word milk and pull that out as distinct from some other word, say cat, for example, so that the machine has the ability to discriminate between something that is the match and something that's not the match. That's the idea of hidden Markov models, when we have a probabilistic way, some variation in what the pattern that we're trying to detect is. The major components of hidden Markov models are shown here, and we'll talk about these in more detail, but I just want to give you a basic idea. So here we have a variety of states being shown. So those are represented by the boxes. And between the states, we have some arrows representing the transitions. So notice that the transitions move from left to right, so it is unidirectional, unlike some of the other kind of models that we've discussed, at least in the standard architecture. And what we have here is we have a series of delete, main, and insert states. And so that's going to characterize the main part of the sequence, the typical way that the sequence comes about. But notice that we have some probabilities in here. This is giving us that idea that the signal could occur by a probabilistic means. So in the main state, we're not necessarily restricted to only having one particular thing, but most of the time, it's going to be, for example, the DNA base thymine. Insert comes from the idea of biology where we would have a multiple sequence alignment, and you may have an event in evolution where you would have a genomic insertion of a DNA base over time. And so this is going to allow to have an insert state where we would have accountability for an insert event that could happen in the sequence. And finally, In multiple sequence alignments, we also have the possibility for a deletion. And so we also have what we call these delete states, where we're going to have this idea that we could have something missing from a particular sequence. And so this is going to be very closely related to multiple sequence alignments of some particular thing. And we're going to have, as we have here, emissions, which are the letters that are expressed in a particular state. So every state can have every letter. So if we're doing a DNA one, we could have every possible DNA in every state. It's just that each of those DNAs will occur with a probability specific to that state. And then we will also have the transition probabilities, which are going to be the probabilities to traverse from, say, for example, the main one state to delete main two or insert two state, and so forth for all the arrows. So this just gives you an idea of what hidden Markov models look like and how they might work. We'll talk about this in more detail as we step through some of the mechanistic parts of the talk. So hidden Markov models can be used to answer three major questions. The first question is the training. How are the emissions and transition parameters of the hidden Markov model going to be estimated from a training set? So as we've talked about in many of our machine learning talks, we'll need to have a training set of data, and hidden Markov models are no different. The next question would be the scoring. How is the likelihood of a sequence computed giving a particular model? So we want to answer the question, how well does this score fit this model? And implicit with that is also an alignment, which is the third question. So the scoring and the model are linked to each other because the score will depend upon which alignment we use. And so there is this idea of having an optimal alignment through the model, that is how it will move through the various states. And what we want to do is we want to find the most likely path of a particular sequence of interest to go through the model, which will give us the optimal score. Why should we use the hidden Markov model? Well, equal importance of every site is not assumed. So for example, what's really useful about this is that every DNA or whatever you are choosing to do, whatever you'll have a collection of features that you're going to express, whatever those options are, the position may determine what the probability of having those options are. So for example, a DNA base may have a probability of a certain amount in a particular position because maybe it's involved in a recognition activity with the side chain of an amino acid and a protein that binds to it. And then the next position may not be involved in such an interaction. And so what's really terrific about hidden Markov models is it allows you to have different probability ratios for the different positions of the alignment to reflect the information that you have available. It's also rooted in statistics, so all of the information that you've learned about Bayesian statistics and so on applies to hidden Markov models. This can be trained on aligned or unaligned data because the training process involves doing an alignment as part of it. There's also variability assumed to arise from genetic mutations of the common ancestor. So this is what we were talking about for the allowance of insertions and deletions. And a point mutation would be represented by having the various probabilities within the same state. So we're going to be talking about hidden Markov models in the context of a particular problem that is of interest in biology, and that has to do with a regulatory protein called cyclic AMP receptor protein, or CAP. And so just to give you an overview of how this paradigm works so that you can understand how this is happening. The basic idea is that the cyclic AMP receptor protein is one common protein, being shown here as this Pac-Man shaped object, and it binds to a variety of different genes, and it causes these to become transcribed. This is a bacterial gene that's well known. And so the point is that this protein binds in all these different promoters. It binds for all these different genes, but the sequence is a little bit different in each case. And so it becomes a really interesting and useful case study to pull out the sequences that the cap protein binds to, because then we can use that to study what the variation in those sequences are. And so one of the research questions we were interested in answering is characterizing what that sequence variation is. So this is a picture of the crystal structure of that protein. It's a homodimer, and it has cyclic AMP, which is its regulator. And this is being bound to a DNA. And you can see that there are some side chains which reach in and recognize specific DNA sequences. Because of this recognition, there are certain parts that are highly conserved. And so this is the representative of a multiple sequence alignment of hundreds of different DNA sequences that the cap binds to. And so these positions here being taller are showing you that there's more of an information content. So this is an information graph in bits. And so what this is saying is that TGA is highly conserved right here, which is corresponding to this section that is undergoing this recognition right here, whereas other positions are less well conserved. And being a homodimer, we noticed that one half site is more conserved than the second half site, because we may have some expectation that there would also be conservation on the other side, which is somewhat more conserved than the rest of the DNA, but not so much. So that will be the system that we're looking at, and this gives you some idea as to what the information content of those sequences looks like. So let's talk about hidden Markov models and probabilities. You may have heard of Bayesian statistics. That is some statistics where we say we cannot actually compute the probability, but we can postulate scenarios and ask how likely that is. So it's, if you make an observation, you roll some dice, we might say, I think that could be a loaded die, because you keep rolling 6 every time. You say, well, that seems a little bit odd. So we might postulate it as a Bayesian question by saying, what is the probability that this die is loaded versus what is the probability that this die is not loaded? And so we can compute those probabilities and then compare between them. This will become very important as we actually look at how hidden Markov models actually work. So, welcome to a slightly dishonest casino, where you may encounter loaded dice. So let's imagine a situation where you have a bag of dice here, and we're going to do the exercise where you're going to reach into the bag and pull out a die, and then you're going to roll that die and observe what number comes up. So these die happen to be in the ratio of 1 to 9, where we're going to have fair dice in 9. Sorry, these colors probably should be reversed. 9. Hopefully it's not this bad. We're going to have 1 loaded die to 9 fair die to give us 10 total dice in the bag. Okay. So if we were to pull out the fair die, we would presume that we would roll each of the sides of it 1/6 of the time to give us 1/6. And if we were to roll the loaded die, this one is loaded such that it rolls a 6/2 of the time, and these other numbers equally spaced. So that'll be 1/10 of the time and 1/2 here. So this is what we would expect from our loaded die, and this is what we would expect from the fair die. And so because these are in a ratio of 9 fair die, if we randomly reach into the bag and pull one out, and we look at it, we would have a 90% chance of pulling out a fair die. So if we have just seen this unfair, this loaded die, we'd have 90% chance of getting the fair die the next time. And if we are doing a fair die, we would have 10% chance to pull out an unfair one. Notice that this works with replacement. So it means you roll the die, you look at the number and record it, and then you put the die back in the bag, and then you reach in to the bag of dice and pull out another one. So it could be possible to select the same one twice. So then we start to wonder how can we use this in order to understand what's happening here. So imagine that we have the situation where we know what the ratios are in the bag, and we know what the probabilities are. Suppose we wanted to make some calculations. All right, well, so in this case, our observation is we rolled 1, 2, 3, 4, 5, 6. And we're going to make a postulate. We're going to, which I'm calling the guest here. So let's say that we're going to say, well, I think it's from the loaded die. Well, how likely do you think that is? So what we're going to do here is we're going to multiply out the probability of the various events, given the fact that it is a loaded die, or given the postulate that it's a loaded die. So when we do this, we're going to use the rules of probability, which say to just multiply out each of the terms. Okay, so we're going to do 0.1 for this transition times 1/10 for each of these, and notice that we have the 1/2 for our last one down here. Okay, so we multiply these all together, and it works out to 0.25%. Okay, well, the other postulate could be suppose that all 6 of these dice were fair. So then we're going to multiply these all together. The 0.9 for this transition here, and the 1/6 for these emission probabilities here. So notice that we multiply the transition probability times the emission probability. So this is the one from the arrow, and this is the one from how many times we actually saw it on the die. And we multiply that out for each of these according to this postulation. And it works out to be 90%. So what this says is that we can't decide with 100% certainty which way it was, right? Because our best calculation gave us 90% certainty, but that's a lot more certain than 0.25%. So this gives us an idea that probably this observation is telling us that this was probably a fair die and was not the loaded die that we picked. The next thing we observe is suppose we actually didn't know what the ratios were of the dice in the bag. Okay, so it's a mystery grab bag. You don't know how many are in there, and you don't know what the ratios are. But you do know that we have replacements. So whatever probability we get for one of them, the other one must be 100% minus that one. All right. So here we're going to make some observations over 100 rolls. Here's 1, 2, 3, 4, 5, 6, and these are the numbers that we rolled. So 6 is a little bit more frequent than these other ones. So we can do this by, again, computing, but instead of knowing what this probability is over the arrows, we're going to represent that by x. And because we know that the probabilities have to add to 100%, I can express the second one as 1 minus x. So we're basically just taking the same formula that we used last time, but replacing in x. So we can solve this. There's one variable, and we can solve this. So x will be equal to 0.9, which is exactly what we had the last time, 90%. And then we can solve for the 1 minus x to get this information here, which works out to be 0.10. So we're able to solve this problem. And that's terrific because that means we can compute this numerically. So being able to numerically solve something means you could use some algebra to solve the problem. So if we don't know the mix in the bag, we can solve that problem by doing this setup as shown here. However, what if we also do not know what the probabilities of the dice are ahead of time? Oops, sorry. This is a much more difficult situation, and so we can see that we would now have more than one unknown. We can't just express this in terms of x and 1 minus x, and because of that, and because we only have a single equation, then we have more variables than equations, and so that's not a directly solvable thing. And so in order to be able to do it, we're going to need to turn to alternate ways of doing this, and this is why we are in fact solving this by numerical methods. That is actually using a calculation, a simulation of a sort to generate the data based on some information. And so what we'll need to talk about then is how do we train our hidden Markov models under such a situation? Because in fact, We don't know what the probabilities are for the various DNA bases, and we also don't know how many choices there are in the bag. So just to represent that in perhaps a more pictorial way, what we have here is we have some bags with dice in them, and we don't a priori know what's in them. I'm representing these dice as four-sided dice because if we're talking about DNA bases, then we have four choices. And so that becomes our alphabet, A, G, C, T. So our alphabet before was the numbers 1 through 6. Now we're going to have A, G, C, T. And so we can have the possibility to select a DNA base at each of the main states and also the insert states. Delete states don't have a bag there because it means that it's a delete state, so there's no base expressed, so there is no emission probabilities, but delete states, only transition probabilities. And so what we're doing is we're picking out of a variety of different bags, which may or may not have the same mix, which is being represented by these bags having various proportions of the colored dice in them, suggesting that it could be different in each one. So then we need to talk a little bit about hidden Markov models with this Bayesian statistics in mind to understand how we might go about solving this problem so that we can get meaningful probabilities into our model and actually do anything with them. So the first thing we'll talk about is hidden Markov model structures. So the linear hidden Markov models are the ones that we've been talking about so far and are in fact the most common, where we start at the start and we're going to proceed through the model through the main states mostly until we arrive at end. However, there would be the possibility to go to the insert states or to the delete states. And as we talked about, that's related to how evolution actually happens. And so we consider that having changes in the sequence could occur by those mechanisms. However, that's not the only architecture that one could have. This represents just a couple of other options we could have. We have tide, loop, and wheel. So in the loop architecture, we have the possibility to break out into this little loop section and do something else in addition to going on main. So that might be, for example, instead of an insertion, it could be an insertion of a whole group of residues. And so this is how one would take that into account. In the tide architecture, you notice that the number of transitions is greater than it is in the standard hidden Markov models that we've been talking about so far. So that allows for a greater variety of allowable transitions. In a wheel hidden Markov model, one is not restricted to a particular number of times through. You can keep going around and around the wheel, and this is very useful when we have a cyclic pattern. This is particularly useful, for example, for nucleosome data. So a nucleosome is a protein that wraps up DNA, and it binds the DNA with a preference. And so the periodicity of that is somewhere around 123 base pairs. And so using a wheel hidden Markov model can capture that periodicity. So you might, for example, align a whole genome to this and ask questions about, is there the presence of periodicity for preferred nucleosome binding sites, and can we identify them? And that's what hidden Markov models such as the wheel have been applied to. So the next thing we need to talk about is how to train the model. That is, how do we get in those probabilities? So far, we've actually just put some probabilities up on the screen, but how would you in fact actually get the probabilities in, especially if we don't know what they are to begin with? So in order to do this, we're going to learn it from a training set, just like we do with many other machine learning activities. You need to have a collection of protein or DNA sequences that exhibits a pattern of interest. So similar to speaking a word as we go around the room, what we might do instead is we might have a collection of DNA binding sites, such as for the cap protein, that all exhibit a pattern that represents that represent sequences that cap binds to, but they are not all identical. Then we can use this to learn the parameters of the hidden Markov model, and then we would parse the training set so that we're going to test some sequences so that they'll be explicitly known to the model during the training process. And then we might have some others that we hold back from the model to see if it's able to find those binding sites, even though it didn't explicitly train on them. The training process will go something like this. There'll be emission and transition parameters that are going to be recursively adjusted such that the sequences in the training set favor passing through the main state. So if you are going through the insert state, often what that represents, more than you're going through main, what that suggests is that your main is not yet long enough. So you could add another state into your length of the model so that it will pass through main more frequently. And then that should go away. So that will help you optimize the size of your model. And also you want the emission entropy to be low, meaning that you should have most of your emissions should be converging on a single thing. So you should see a pattern that is growing out of this. You shouldn't have all of the states be 50-50 for everything, because you wouldn't really have a pattern then. So let's just talk about a simple case of how you might have an emission parameter estimation. So in this, the simplest thing we could think about, we would have some sequences. So here are 5 sequences. Let's say that these are cyclic AMP receptor protein binding sites. So this actually represents the contact region of that, which is reasonably highly conserved. And so what we have here is the first position, the second, third, fourth, and fifth positions. And what we're going to do is we're going to calculate the probabilities for A, G, C, and T. So a reasonable way to estimate those would be to simply just count them up and calculate their percentages. So I made it pretty easy because there's 5 of them. So we can more or less do this in our head. Since in this case we have the appearance of 1G, this means it's 20%, and the others are going to be represented 4 times, so that's 80%. So we can say that these are our probabilities in the model right here, just based on our observation. We're going to have similar probabilities in the second position because we have 4 Gs and 1 C. So we've just moved those to the appropriate positions. The third position is 100% conserved at having thymine in all of them. So we're going to have 100% at T. The next one is 100% at G. And finally, we have some increased variation in the last position, which has 20, 40, 60% of A, and then shared 20%. It's separated between the G and the C, which we can see right here. So that's pretty easy. This is how we could calculate this in a very simple case where we have no gapping and the pattern is really straightforward to see what it ought to be. But we could do this in a bit more of a different situation. And so what we're showing here is that what we're going to do is we can actually align those sequences here. So here the main states are being shown along the bottom, the inserts are being shown here, and the delete states are along the top. So when we put the sequence in, what we want to do is we want to align each position to each one of the states to maximize its probability based on what we've seen so far. So for example, we might start with the probabilities that we showed previously and then lay down some additional sequences to match up, and then ask the question, how did it work? So for example, in this case, it's pretty straightforward. These are all having a transition of 1.0, so there's no possibility to go anywhere else. But here it starts to get a little bit interesting because we say, well, it could go up to this insert state, or it could go over here to the next main state. So let's say that we have the question that we want to put down a T next. Okay, so we take a look at the choices of where we could go. We could go through this arrow at a 0.1% transition probability. Let's do this one. So here we have, yeah, so the probability should add up to 100%. Sorry, these are not exactly lined up over their arrows. Yes, this is a self-transition, sorry. So here we have the possibility to transition either from this main state over to here at 0.91, which means we're going to have a transmission probability to go up to the insert state. And so all the possibilities to transition out of this state should add up to 100%. So clearly, going this direction is going to make the most sense. So what we want to compare is, does it make most sense to proceed over to the next state, or do we want to transition to another state over here? So for example, if we are going to put down a G, we see the G is very rare here. It's very rare to observe a G in this position. However, it's very common to see a G in this position. The alternate thing we could consider is we could consider going up to delete and skipping it all together and then going back down here and having a G over in this position. So we need to then consider those different possibilities, and we could compute the probability for those various things, just like we did using Bayes' theorem to calculate the probability of rolling the dice, well, the postulate that the dice were all fair versus the dice were all loaded. And so we can see that it's going to make the best sense probably to put the G into this state here, right? So we would be comparing the probability to do this transition and that emission versus going in this direction. And so what we'll do is we'll calculate up all the probability possibilities, and we'll see which one is the best, and that's how we'll decide where to put it next. So during the training phase, then what we're going to do is once we do an alignment to a particular position, we tweak the probabilities that are in the state. So the thing that aligned there will cause that probability to go up by 1 unit and then by 1/3 decrease in the other 3 so that it will maintain 100%, and so that you will enforce the pattern that's been observed. Now the training rate is going to be how big of a change that you allow to happen when something aligns. So that is another parameter that does get tweaked during the training process. But usually you want something that's going to be some small percentage, say around 5 or 10, depending on your training set size and your confidence in the data. But that's something that gets tested by trial and error as you do your training. So as we do this, we should get a decrease in the training error. Now, the way that the training error is going to be computed is we're going to make use of those sequences that we set aside, knowing what they were, knowing whether or not they're a binding site. And so we can ask the question, how many of those does it get right, having not seen them? And as we iteratively align the sequences and allow them to adjust the parameters in the hidden Markov model, that training error should be decreasing. And as this asymptotically settles down, so you see that more training steps are not really helping much, this is how we reach convergence, and so we don't really need to continue training the model any further because there won't be any expected benefit to that. So this will keep track of how many training iterations, which will again depend upon that parameter, that how quickly we allow the model to update, will then allow you to train your model and know when you've reached convergence. So some things that would be good for a hidden Markov model to do, so to know that your hidden Markov model is good, is you would want to look for things that are going to replicate known trends. So for example, if we knew that the cap protein recognizes a DNA sequence that has a consensus of TG-TGA, it's great to see that we have those recognized and that they have very high probability here, which provides us with confidence that our model is actually working correctly. So finally, the next thing we want to talk about is the sequence alignment and scoring. So as I mentioned before, they are in fact intertwined concepts because the alignment is going to determine the scoring. So when we're doing a scoring, we're doing almost exactly the same thing that we were doing when we were doing the training, but the major difference is that we don't allow the probabilities to update as we go through. So for example, suppose we were going to score up a particular cap sequence. So let's say it's going to be the small e, which is a particular sugar. And so the sequence that we have for this is going to be TG-TGA, and then we're going to have to see what will happen next, TG-TGA, TCT, okay? And so what we're going to do is we are going to align this. So the emission probabilities is going to be 0.8 for this sequence because we look in the first state, it's 0.8. Then the G emission probability is going to be 0.94. The next one is 0.95 from here, so it's basically falling right along the consensus. G is the only choice here, so that's a 1.0. And then A also is highly conserved. And now we have another T. So this T could either go in this position here, with 0.57 probability, or it could go over here with a 0.3. So we see that this is greater. So we're going to assume that this one has an insert event. Okay, so we put that up there. And then the next thing we have is a C. So we're back on track here. And then a T is the final letter. So we see how we're tracing out that path through here. Now we also have the associated transition probabilities, which are the numbers that are over the arrow for this path. So we have 1, 1, 1, 1, 1, and then it starts to get interesting here, right? So this is going to be the 0.3 and the 0.55 as we go up into that insert state and come back. And then the 0.87 is going to come from this right here, and then the probability to transition to N to stop the model, and so that's 0.97. So what we do is we simply multiply all of them together, and we get our score. Now, the issue with that is that when we're multiplying a series of very small numbers together, we may run into a precision problem because, as you can imagine, those numbers are going to get very, very small, and so we're going to encroach upon the ability of the computer to actually store enough number of significant digits. So what we'd really like to do is we'd really like to move into a space that will get away from that. So as you might remember from calculus or pre-calculus class, we could move the multiplication of events into the sum of the log of the numbers. Those are going to be equivalent operations. And so the division of a numerator by a denominator is going to be subtraction by the law of logarithms. So the log odd score is going to be the probability of the sequence over the null model, which is, so since there's four DNA bases, that's going to be 0.25% up to the power of the length, right? So you raise it to a power, you have the number of positions, you raise to the power of the length to get the probability. So this is numerically equivalent to doing the subtraction of log of P of S, the log of the numerator, minus the log of the denominator. So here we have also applied the law of logarithms to bring our L down out in front. So this is how it's equal to. Then we can substitute in our numbers and calculate out our score in log space instead of probability space, which makes this much easier and more accurate to work with, which is why oftentimes when we're working with hidden Markov models, the answer will be reported as log odd score rather than the probability directly, but they're directly related to each other by this formula. So then let's just take a moment to look at some possible outcomes for a hidden Markov model. So these are some different data sets that were used to train some hidden Markov models. And so what we're interested in looking at here is which one is the better model, okay? So in this case, our training set scores much lower. These are log odd scores, has much lower score than other known binding sites. This is our training set here divided into the ones that were actually used for the training and then ones that were not known to the model. And so this is a sign of a poor Markov model because the issue is that it's overtrained. It knows a lot about the sequences that it saw, but it cannot extrapolate. Now the non-binding sites are known other sequences that do not have a cap binding site, which we know from experiments. And so we would expect these to have a much higher score than the training set ones. And so what we see is that the other binding sites, which in fact are binding sites, score almost as well as the non-binding, score almost similarly to the non-binding sites. So this is not a good model because we don't have good ability to do predictions. Here is a model that was parameterized up in a different way, where our training set is performing similar to other binding sites, and most of these similar binding sites are having a lower score than the non-binding sites, which means we have better discriminatory ability of the model, which gives us increased confidence that it's actually working. Another application we can use with hidden Markov models involves doing alignments. So what we could do is we could take a genomic piece of DNA and we could feed it into the Markov model and we could move it along by one base each time we go through and do a scoring. And so this is what is known as convolution, and we can use this then to identify the location of binding sites. So we'll see that that will have a maximum where the score is the best. So we might surmise that there is a cap binding site somewhere in this area here where we have a peak and that there are not cap binding sites in these areas. And looks like there's a second one that might be growing in over here. So we're able to do a wide variety of things with the hidden Markov models. We can use that convolution approach to score up the entire E. coli genome. So we've done that with a couple of different ways of doing it. We've done it with the AGCT DNA base that we've been talking about. We also did another model where we did the dinucleotide steps. So which basically is going to look at the space between the nucleotides instead of the actual nucleotide itself, which changed the alphabet size, which was somewhat of an improvement. Here we're exhibiting a different model where we are calculating the hidden Markov model in a slightly different way. And here is another one that's scoring on some flexibility parameter instead. So basically what we've done here is we've tried the plain DNA, and then we've also switched the alphabet here. And in these two cases, we have introduced some alternate models for parameters besides just the sequence directly, but numbers describe the actual geometry of the DNA bases, which is going to give us some idea as to what those hits are. Now this is reporting on the number of scores that fell within a particular range. So what we want to see is we want to see how many scores are really going to work, right? So we would use the information from that respective model's training and test set to see what a typical score looks like, and then we'd see how many scores we find. So the important thing about this is that as we change the alphabet size and the parameter that we use, we're going to get fewer and fewer sequences that are going to have significant scores. So a score range for a particular binding site should typically be over on this left side, and we see that there are many that have binding sites here, and then it decreases, and these other models which are showing that these other ways of studying the parameters can decrease the number of hits. So we have some outside information on how many binding sites exist in the E. coli genome, and it's not as many as this model suggests. So these ways are going to then suggest ways that we could change up what parameters we use for our hidden Markov models in order to make them better suited for doing genomic scans. So then, today we've talked about an overview of hidden Markov models, getting an idea of what they look like and what they can do. We talked about hidden Markov models and their probabilities at the slightly dishonest casino. We have looked at the architecture, training, and scoring of hidden Markov models, and finally rounded that out with a few results from some hidden Markov models from the literature. So this is some recommended reading and some of the places where you can read about some of the results that I showed for you today. So definitely recommend you have a look at that. All right, great. Well, I hope you found this interesting and useful for understanding how hidden Markov models work. I put together a tutorial for you in order to work with hidden Markov models using scikit-learn. It's based on a web tutorial. And so I hope you will enjoy using sklearn as one of the best softwares out there. It is the basis of TensorFlow, and it has all of the rudimentary code that's required for actually running hidden Markov models. So what you'll be doing today is learning how to use that software and how to go ahead and do some learning with hidden Markov models. All right, thanks very much. I hope you enjoyed today's talk, and good luck with your hidden Markov models. Thank you so much.